Hello, students. All right, today we are going to continue with 11.1 vaccination, and here's the essential understanding. Vaccines contain antigens that trigger immunity but do not cause the disease. So remember that we said that an antigen was any foreign substance that our immune system does not recognize. So vaccinations are able to induce long-term immunity to specific pathogenic infections by stimulating the productions of memory cells. And remember that the ability to remain with that long-term immunity is the persistence of those memory cells. So what is a vaccine? The definition that they give you here is the, the older definition of a vaccine, which is a weakened or attenuated form of the pathogen. We now with COVID know that instead of only having weakened or attenuated forms of a pathogen for a vaccine, now we have genetic vaccines. We see with COVID-19 that the COVID vaccine is an mRNA vaccine. But this section of IB covers these forms of weakened or attenuated forms of the pathogen. And they contain antigens, but they are unable to trigger the disease. So they're not going to make you sick. So how is an attenuated virus made? So there are certain antigenic determinants in a vaccine that may be conjugated to an adjuvant. And an adjuvant could be something that functions to boost the immune system. So I'm going to quickly move here to several different adjuvants that are used in the U.S. It could be some form of aluminum, it could be some form of a lipid, it could be some form of an oil and water. So all these are different forms of adjuvants. And what these adjuvants end up doing is that they help, one, conserve the attenuated virus and also help the immune system function to the antigen that is being presented. So the body is going to respond to an injected vaccine, and that's going to start a primary immune response, which results in the memory cells being made. So in the future, if you get exposed to the actual pathogen, the memory cells are going to trigger a more potent secondary immune response. And that means that the disease and the symptoms are not going to develop and you're thought to be immune. Now, importantly, the length of time a person remains immune depends on the persistence of the memory cells, right? So sometimes you're going to require a booster shot to maintain that particular immunity. So here we see how vaccinations work, right? We see the pathogen here, and then we see how the pathogen becomes attenuated, and that means that the scientists are going to take the actual pathogen and they are going, it depends, because different vaccines work out differently, but sometimes they might remove, for example, a particular portion of the antigen, and then they insert that particular portion of the antigen into a less virulent virus, for example, an adenovirus. So they might take this antigenic determinant from the actual pathogen and they attach that to an adenovirus, a lesser form of a virus. So it's not going to make you sick. And then they add the adjuvant, and then that's pretty much what the vaccine is. And then they inoculate you, and that is going to trigger your B cells to make plasma cells and antibodies, but also to make these memory cells. And these memory cells is what's going to give you long-term immunity. So how does herd immunity work? So we pretty much now know well the definition of a pandemic, right? So an epidemic, as opposed to a pandemic, is where you have an occurrence of a particular infection within a given region, so it stays localized. We know that a pandemic is an epidemic that has spread across a large geographical area, like a continent. So this is what we're seeing today with the COVID pandemic. So, how do vaccination allow for herd immunity? So, herd immunity is when individuals who are not immune to a pathogen are protected from exposure by the large amounts of immune individuals within the community. So, sometimes people cannot get vaccinated because either they're too young or they have a compromised immune system. So, what happens when the majority of people are not immunized? Well, here you see in blue that there are susceptible people amongst the population, and then you see the contagious people, right? So, when there is no immunity, you see how the people who are contagious, they are able to spread the virus amongst all the people who are susceptible, or pretty much the majority of them. So the infectious agent passes freely from contagious to susceptible. But when the majority of people are immunized, this is what we see herd immunity. We see that the contagion or the virus cannot pass freely via the immunized people. So here we see the immunized people in yellow. Here we see the contagious people, and then in blue the susceptible. So those people that are immunized are providing that pretty much blockage of that virus to pass from one person to another. So then the susceptible people are indirectly protected by the people who are immunized. So here you have an application, which is a little history on smallpox, which was the first infectious disease of humans to have been eradicated by vaccination. So we know that, what do we mean by eradicated? That means that the disease stops circulating. And if it stops circulating in a region and then worldwide, we consider it to be eradicated. So smallpox was targeted for eradication in 1967 by the World Health Organization, and they started a global vaccine program. And we know that the last known case of smallpox in a civilian was in 1977, and the disease was eradicated by the World Health Organization in 1980. Now, there's reasons why smallpox was targeted and why it was successful. So smallpox is easily identifiable due to overt clinical symptoms. So you can tell when a person is infected. For example, you see the blisters, right? So you can tell. Transmission only occurred via direct contact, so the person has to pretty much touch you. So there are no animal vectors or reservoirs. The infection period was short-lived, 3 to 4 weeks, and the virus was stable and didn't mutate into alternate strains. And there was global cooperation, and immunity was long-term, so there was a persistence of those memory cells. So repeated booster shots were unnecessary. So that was, it kind of all worked out for the eradication of smallpox. So as you can see the timeline here of the eradication. Here's a good TED Talk of how we conquered the deadly smallpox virus. And then the skill here, we're not going to spend much time going over this, but this is just how you're able to analyze epidemiological data related to vaccination programs. So there is a website here for the WHO data and statistics, and this is where they keep records, and it can be accessed by anyone in different countries. Here's also some vaccination data in Excel format, and it shows you data tables and graphs of the different national vaccination programs. And here is just an example of a data table that shows you the incidence of measles in Australia based on the WHO data records. So you can, for an IA or database analysis, science fair investigation, you can take these data tables and you can analyze them and see if you can find trends with these different diseases. Okay, so that is it for today for vaccination.